Hi again, everyone, and welcome to the third session in our T-Cell Talks event. I'm Liam Sanyo from the events team here at scientist.com, and I'll be hosting this one. With us now is Dr. Julian Lum, distinguished scientist at the Dealey Research Center at BC Cancer, who will discuss key factors to consider when choosing metabolic assays and how they can be used to assess cell function and cover metabolic characteristics of different commercially available cell culture media formulations. So with that, I'm very pleased to welcome Dr. Julian Lum. Julian, thanks so much for joining us today, and the floor is yours whenever you're ready.
Okay, great. Thank you so much, and I just want to start off by thanking Stem Cell and the rest of the sponsors who are putting on this wonderful event. Before I start, I'd like to just acknowledge the people that I've worked with over the years, in particular Sarah McPherson, whose work I will present today, and a number of collaborators across our metabolomics, clinical, our immunotherapy, biospecimen team, and especially our bioinformatics team. I also want to highlight ongoing collaborations I have with Stem Cell Technologies and colleague Jason Cantor. And of course, all of the sponsors and funding agencies who've made the work that we're doing possible. Here are my disclosures, and I just want to highlight that I will be talking about some results and data that was generated in collaboration with Stem Cell Technologies. Okay, so I think it goes without saying that CAR T cells in the space of cell therapies has really demonstrated remarkable success, particularly in the hematological malignancies. And as you can see on the left, in this paradigm of generating these cells that can be used as a living drug to treat patients with various hematological cancers. And to date, there are 6 CAR T cell approvals. There are over 1,675 clinical trials employing CAR T cells in the oncology sector. And more recently, there's been a lot of interest in deploying CAR T cells in the setting of autoimmunity. And at present, there are over 70 clinical trials that are exploring this new possibility. And it's estimated that the current market value for CAR T cell trials is in the order of about $3 billion. So a tremendous amount of biotechnology investment into this sector. Okay, and so I'd like to just highlight also that the manufacturing process, in Canada, has really come from the inception of an academic endeavor, and there are 5 centers right now that are either operational in terms of point of care manufacturing or that are currently in development and hopefully will be deployed shortly to be able to increase the capacity of point of care cell therapy manufacturing across the country. And here I've just highlighted some of those sites and where they'll be set up. Okay, so for today, there are 3 things that I would like to go over as far as the delivery and the material. And the first one is really the considerations for what type of metabolic assay that one would use to interrogate the features of a given immune cell type, and how can we use that to assess the functional features of those immune cells. And the second one is looking at now primary data, which I'll present to you on the metabolic characteristics and the impact of different types of GMP-grade media formulations that are being used to produce and manufacture cell therapies, and how do those alter the metabolism of the T cells, and can we use them in a way that retains the metabolic fidelity of those cells that are being produced. And then finally, looking at how we assess those media products and what are the metabolic features that we can measure during the manufacturing process in that particular scheme. Okay, so one of the things I get asked a lot from colleagues and others is how do I measure metabolism in T cells? And I'm just going to very quickly go through three, I would say, of the major workhorse assays that are being used. And the first one is the Seahorse analyzer. And there are a number of advantages of this particular instrument, and I won't go through all the details of this, but suffice to say that it's a very easy plug-and-play system where you need very few cells that really give you outputs of oxygen and extracellular acidification. And some of the limitations are that the outputs are often on bulk cells, although one can do some pre-enrichment steps to look at cell type specific metabolism. And really the key point I want to make is that the Seahorse is really looking at two parameters, oxygen and glycolysis. Using multispectral flow cytometry, of course, now you're able to get on a per cell, single cell basis, you're able to quantify specific cell types based on the panel of markers that you might use. So you're enriching the depth and the repertoire of information on a per cell basis. The limitations is that they often are single metabolite or metabolic readouts looking at one particular pathway, although there are some new algorithms such as CNIF and others that now you can actually do more sophisticated pathway analysis and modeling. And finally, the other limitation is that because this is an antibody-based system, there's a fair amount of upfront optimization that is required. And then finally, there's high-resolution mass spectrometry. And really, this is where you get a large breadth of metabolite identities on the order of tens of thousands. You can also distinguish between isomers because we know isomers have different roles in contributing and regulating immune cell function. This is a highly sensitive and quantitative assay. And some of the limitations is that, in order to get this breadth of metabolites, you do need a fair number of cells. And you have to worry about high turnover metabolites that might not be able to be detected simply based on those properties. And so some pre-processing sample preparation is often required in order to capture some of those. And then in terms of cost, the cost does increase both on the complexity of the instruments required and some of the consumables that are required for these techniques. Okay, so now I'd like to turn my attention to actually some data that we generated in collaboration with Stem Cell Technologies on looking at various media formulations. And I do want to highlight that we are using the Clinimax Prodigy system here in Victoria as the main platform for our cell therapy manufacturing. And so, we're going to focus, at least I'm going to focus on kind of the ex vivo approaches that we're using that would allow us to reverse engineer onto the Prodigy system in terms of manufacturing. Okay, so here I'm going to really highlight the work of Sarah McPherson, a research associate in my lab. And one thing I want to point out is that the data that I'm going to present today is really meant to be a broad overview of characteristics and not an endorsement of any particular platform, because I think depending on the user's requirement or needs or application of the product, I think that really is an important consideration in terms of what platform they'll use. And so we tested 5 different platforms listed here in the first column. I've abbreviated them and you'll hear me refer to them using these abbreviations, you'll notice that the activators and stimulators that are being used are quite different across these platforms, as are the media. And if you just simply look at two metabolites, glucose and glutamine, you can already notice that there's an obvious difference between the levels of these two particular metabolites in these media formulations. And so that's a real, I think, motivation for us to understand how these different concentrations may affect the metabolism of T cells. So here what I'm showing you are healthy donor T cells, which we've activated in their respective media using their respective stimulators, looking at the number of cells that accumulate over an 11-day expansion ex vivo. And what you can see is there's a wide range, anywhere from 40 to almost 600-fold expansion of T cells. These are isolated T cells, and you can see that, one of the things I'll point out is that on day 4, you can see that the trajectory of the cells appear to be maintained over the remainder of the expansion. And what I mean by that is that these lines don't actually cross at any given point, which kind of suggests to us that the metabolism in the media using these activators are programmed in such a way that they persist throughout the time course of the expansion. And then when you look at the ratio of CD4 and CD8 positive T cells, you can see that at least at the beginning, there's really no difference between the ratios over the different stimulating and media conditions. However, at the end of the 12-day expansion period, the ratios of the CD4 to CD8 appear to be different depending on which media formulation. And that's an important consideration because there's literature that suggests that the ratio of 4s and 8 may have some impact on the efficacy of at least CAR T cells in the setting of CD19. We also looked at the mitochondrial activity using a flow-based assay with MitoTracker Deep Red. And what you can see in this plot here is that there are quite variable differences, of mitochondrial activity. The rapid expansion protocol appears to have the highest, and this changes depending on which formulation and which activator system that you do use throughout this expansion period. The other is the generation of superoxide as a result of the changes in mitochondrial activity of the cells. And here I'm just showing you mitosox red on the y-axis and the mitochondrial mass on the x-axis. So you can really see that the mitochondrial mass doesn't change really significantly, but the percentage of cells that are producing superoxides does vary quite significantly depending on the type of stimulator and media that is being used in this expansion. Okay, so the next thing we wanted to ask is whether the stimulators, the activators themselves, play a role in the metabolism or altering the metabolism of the cell. And so what we did was we fixed the media using this CTL-AIM5 combination, and we stimulated the T cells either using the ICM stimulators or the TAC stimulators. So the media is fixed, but the stimulator is variable. And what you can see here is that, similar to what I showed you in the previous slide, it doesn't appear, the fold change in cell number appears to be similar across these three conditions, and does not appear to change based on the activator that is being used. This is similar to the previous slide that I showed you. And we also don't really see any difference in the mitochondrial mass when we swap the different stimulator conditions. And if you look at cytokine secretion, there's really no change in the amount of cytokine produced on a per cell basis when you swap the different stimulators. However, now, if you do the reverse experiment where you fix the stimulator, either using ICM here on the left or TAC on the right, and you change the media in which the cells are being stimulated. So we looked at the 3 different medias, the CTLA-5, the ICM, or the TAC. Now what you can see is that there are actually quite substantial changes in the accumulation of cells depending on the media that you use, okay? And so you can see both using the ICM stimulator or the TAC stimulator that there are profound differences in cell number. Similarly, if you look at the mitochondrial mass, you see also these differences. So these 3 here are the different medias, and the bottom, this set is stimulated with ICM activators, and this set is stimulated with the TAC stimulators. And you can see here that depending on the media that's being used, you can see that there's differences in mitochondrial mass, as well as differences and trend towards differences in the amount of cytokines that are being produced by the cells in those respective medias. Okay, and similarly, what we did was we activated cells in their respective media and their respective stimulators. So that's denoted in the solid color bars here. And this is over a 12-day period of stimulation. On day 12, we switched the media. So from ICM to CTLA-5 or from TAC to CTLA-5, etc., in these hatched bars here. And what you can see is that when you switch the cells after they've gone through a 12-day expansion into a different media, the CTLA-4 media, you can see that in some cases there was actually a dramatic change in the percent of interferon gamma or effector cytokine secretion, depending on which media that you use. That's true for both interferon gamma, but also TNF alpha. And you can see that there's actually an improvement in the cytokine secretion when you switch them into this different CTLA-5 media. So the media formulation appears to have a much bigger role in altering, or at least dictating, both cell proliferation, mitochondrial mass, and also cytokine production. Okay, so I'm just going to then conclude and highlight that it's really important that how you determine and what assay you use to actually assess metabolism of T cells really is dictated by what the question you want to ask. Okay. Secondly, it's really important that you understand what each one of these assays can measure for you, so that you know that the results of the data allow you to make conclusions based on what the assay is measuring, whether you're measuring an absolute abundance of a metabolite or where you're actually able to look at where the metabolites are coming from. One of the things that I think has really illuminated us based on just this previous work that I've presented is that it appears that we can uncouple metabolism from the function of the cell, right? Because when we switch cells from different media formulations, we were able to see a very distinct change in cell proliferation and as well as cytokine production. So that's an important consideration when thinking about, do we want a cell that's more proliferative, or do we want a cell that's more functional from a cytokine perspective? And we think that the formulation, the constituents in the media, is going to be a really important contributor or driver of how that cellular product behaves, not only during the expansion, but at the end of the expansion, and then how that cell will be actually used in an application or therapeutic setting. Okay, with that, I'm happy to stop here and be delighted to take questions. Thanks very much for your time.
Excellent. Well, thank you, Julian, so much for the great presentation. And with that, we'll move right on to the Q&A. So, Julian, first question here. So do you foresee point-of-care engineering becoming the new normal in industry and clinic? And can you talk about some of the obstacles when transitioning from academic centers to clinical application?
Yeah, so thank you for the question, Liam, and thank you everyone for joining in the session this morning, at least morning on the West Coast. So I think that I'm going to answer the question in two sort of ways, and I really didn't speak too much about kind of the logistical aspects of a centralized system versus a point of care kind of a platform. But, I think there are lots of advantages and disadvantages of both. And, really some of the pain points that we have to consider, not only on the top, in the area of the assessing the metabolism or some aspect of the media formulations that we use to generate cell therapies, in the setting of autologous manufacturing is really when we're talking about high out of specification and manufacturing failure rates. I think we need to think about that. Are we using the right ingredients, if you will, to ensure that we can mitigate that those kind of failures in the manufacturing? There's also a lot of complexity depending on what jurisdiction that these cell therapies are made in terms of the viral vector specifications. In whether you're in Canada or in the U.S., and I think at least presently we're still using viral vector systems to create the cell therapy products, particularly the CAR T cells or the TCR engineered T cells. And so we have to think about that. And then there's also the consideration of the length and the variable lead times it actually takes to do all the patient coordination, right? Those challenges from where is the patient located, where are the cells being made, where is the patient going to get their infusion. So those are also, so that is also an important consideration. And then that could potentially lead to, missed sort of opportunities, therapeutic windows where you can actually, sort of deploy. And, at the current sort of market rate, the centralized systems for making CAR T cells are on the order of, $250,000 to say $400,000 per batch. And a lot of this cost is really around sort of like the infrastructure, the assets that are used in a centralized facilities to make this. And so, moving away from that system to a decentralized system, I think there are some important considerations because of the manufacturing logistics. In a decentralized system, there's flexibility to streamline certain processes that could lead to sort of faster vein-to-vein times. And then the cost structure, I think, also kind of shifts towards a more operating expense as opposed to that big capital cost of a large facility. And so there's some savings around that. And the other thing I want to point out is also in a consideration of decentralized versus centralized, there's the cryogenic logistics, right? You need leukapheresis products, then you need to send those products to different places. And so those are really important considerations. Are there sufficient T cells from a leukapheresis product? And so the decentralized system does offer some advantages because there's the sort of agility to do a restart very quickly if things you run into problems. And then I would also say that the which is leads me into the comments about the products, the things we're using to manufacture the T cells, which is really the local sourcing of materials, of all of these materials. And we're on the West Coast at least, having stem cell technologies and other players close in the local ecosystem, I think really helps. And then I think the one thing I really want to highlight, because it is such an important part of any center that is manufacturing cell therapies, is talent and partnership. The labor, we create training centers where you can really attract and retain highly skilled workers and help the economy in many different ways. So I think there's really lots of benefits of that kind of system. And I think moving forward, the ability to innovate in using new technologies. Of course, we've all been aware of the CRISPR engineering side of things. I think that will really begin to shine, and having local capacity and capabilities to do sort of iterative cycles of innovative modifications, I think will be something that the field will continue to move towards.
Right. Yeah. Great answer. Yeah. Some really, really good points there. I think in the interest of time, we'll just have one last question, Julian. And so what kind of media properties do you consider when you're choosing ancillary reagents?
Yeah, this is a really great question, Liam. And I think I really want to highlight that as cell therapies begin to branch into other indications beyond oncology, but even within oncology itself, because that tumor microenvironment is so heterogeneous. And I think the considerations of the media property really depends on, what's the tumor environment these cells are going into? Or is it going into a different setting like autoimmunity? Or, there's some studies looking at using this in sort of the cardiac field. And so I think it's not a one size fits all, and it's really going to need quite a bit of tailoring depending on the product and its application. And I think that's really the take home message that I want to leave everyone with, is what you, I'll say, feed your T cells with, or any other immune cell with, is going to be highly dependent on the application. And I think that's where being able to have in-house capabilities to manufacture, do optimizations specifically for the purpose of that particular cell therapy is really crucial. And I think it's really important that each center, depending on what they plan to use the cell therapies for, be able to test that and be able to do that in an iterative way. So there's no one size fits all, unfortunately, but hopefully there will be continuation of different products on the market that people can use and test.
Fantastic. Well, we'll leave it there, but thanks so much, Julian, for your time today and for the presentation. It's really been great having you with us.
Thanks very much for your time.